This is a production of Cornell University.
So, as was mentioned, I just recently started my assistant professorship at Penn State University. And so all of this work that I'll be telling you about is all work that I did previously at the University of Arizona, but will be continuing on in my new position. And so we'll be looking at the causes and consequences of antagonism among different isolates or different strains of Pseudomonas syringae, and we'll be focusing in on this molecule here that I'll be telling you more about, or this structure, I should say. So first, there's a lot of people that have been interested in this field of thinking about plant-associated microbial communities and the different factors that influence these communities. Of course, these have a number of effects for plant health and production. This crowd is, I don't need to explain that in detail for this crowd. But there's been a number of things that have been, over the past decades that have been understood in terms of plant host being important, which organ you look at, or even more recently, what the plant genotype is in terms of determining what the microbiome for the plant is going to be. As well, there's also been this understanding that the other microbial members that are present are helping structure the community as well. And so this is really where my interest focuses, is understanding the interactions that are occurring between these resident microbes within the plant environment. And so my research questions really focus on how do microbial interactions affect the ecology and evolution of these plant-associated microbes? What are the genetic bases of these interactions? This is where I focus, or where I come from a very molecular genetic and molecular biology background. So I'm always interested in what the mechanisms of these interactions are. How these interactions are then going to influence downstream plant microbe interactions. And then finally, as was alluded to, how can we exploit these interactions for say pathogen control or stimulating communities that are going to improve plant health and productivity. But for the purpose of today, we're just going to focus on these two areas here. And so a lot of people here are very familiar with Pseudomonas syringae. I won't belabor it too much, but it's a ubiquitous phytopathogen. There's over 50 or 60 different pathovars now that have been described. And so as a species, it can infect a wide range of host crops or host species. And so when we think about the phases of bacterial-host interactions, there's a lot of work, a lot of great work done here thinking about the pathogen once it gets inside and sort of the interaction between the microbe and its host plant at the genetic level. I come from the background coming out of Steve's lab at Berkeley, where the focus was a little bit more on understanding the microbes as they're existing on the leaf surface. And so what are the stressors that they encounter there and how do they survive there so that they can build up a sizable enough population that ingress into the plant host is a likely occurrence. And so we know that things like acquiring nutrients, evading desiccation stress or desiccation tolerance, as well as tolerating UV exposure are all major factors for bacteria living in this environment that they have to tolerate. So when we think about the microbes then on the leaf surface, one thing is that we need to realize or keep in mind is that they're found in aggregates. So they're not spread evenly across the leaf surface, and that these aggregates tend to associate with particular anatomical features. So the base of glandular trichomes or in the intracellular grooves, and so that's what I'm showing here. With this glandular trichome here, we have a large aggregate of bacterial cells here forming. And there's a number of reasons why we think these aggregates form at these locations, and one of them is that the data seems to suggest that these are really where you're getting leakage of water and nutrients from the leaf interior onto the exterior. And so this is where the bacteria are actually going to make a living. And so if we think about these microbes fighting for finite resources, of course, there's going to be competition. There's only so much space and only so much nutrients that are leaking out. And so we think about, we know that there's a number of different species, and within a species there's a great, especially for syringae, there can be a great diversity of different strains that are inhabiting the same environment. And so we think about them potentially interfering with each other's ability to compete. And this could either be through acquiring resources more effectively than the other member, but the thing that I'll be telling you about today focuses more specifically on antimicrobial mediated antagonism. And specifically, this class of compounds here referred to collectively as bacteriocins. And this is a large group of different proteinaceous compounds. And you can think of them essentially as highly specific antibiotics. So with an antibiotic, you would expect that most all members within a given species, if that species is sensitive to the antibiotic, most all of the members will be sensitive to that same antibiotic, except for those that have a specific resistance cassette. For these systems, it's quite the opposite. Just because one strain of a species is sensitive to a given bacteriocin, there's no reason to expect another strain will also be sensitive. And so there's a high degree of specificity, again, and they're also protein-based. And so this is another thing that separates them from traditional antibiotics. And this is a little bit of a renaissance within this field, I would say. There's a number of groups that have started to become interested, not just within syringae, but within other pseudomonads, other environmental pseudomonads as well. But if you look at the literature from about the mid-60s through the early 70s, there was also a number of groups at this time looking at it. Ann Vitiver's group in particular is one that I'm just highlighting here, these bacteriocins. But at this point, all of the tools or all of the questions that were being asked were using bacteriocins basically for species delimitation as well as epidemiological markers. And of course, the tools available at that time didn't allow for really in-detail analysis of the particular bacteriocins being produced. And so the field shifted away from this into a number of different angles. And so this is sort of something that, again, had been looked at, but there's still a number of very relevant and important questions that need to be asked. And so the three things that I'll tell you about today is the genetics of bacteriocin production. This is all, again, within Pseudomonas syringae, as well as some of the work on the genetics of sensitivity, and then some of our hint as to what potential ecological role these actually may be playing in the environment. So first off, to screen for bacteriocins, it's very straightforward. You just have your strain growing up in a broth culture. You add some chemical inductant, or you just let it grow long enough, and eventually the cells in there will lyse and release the killing agent, in this case the bacteriocin, into the supernatant, filter sterilize, pellet the cells, and then just you have the sterile supernatant here. And then you spot this onto a lawn of recipient bacteria. And so in this case, the donor strain is going to be one Pseudomonas syringae, in this case probably pathovar, and then all of these recipient strains again are going to be different strains of syringae. So as you would expect, if you just had a medium and you spotted on sterile media, you won't get any inhibition of growth. But with the supernatant containing the bacteriocin and a strain that's sensitive, you'll get a very defined killing zone. And as I mentioned, you can have the same supernatant on an alternative strain and you won't see any inhibition whatsoever. So they're very specific. So these were the original 18 strains that I selected for screening, and this represents a fair amount of the diversity within Pseudomonas syringae, although there's many more environmental strains and other phylo groups now that we know about. And so this is showing just the cladogram of their relationship. And all of these columns are going to indicate where the activity is coming from. And so I'm spotting all by all. All of the columns are going to indicate where the activity is going. So these are the producers and these are the sensitive strains being tested. And so where there's a white box, that'll indicate killing activity, and a black box will indicate no activity. So the first thing you can see is that there's actually quite a diversity of killing activity among these different strains. And in fact, you can see that there are certain patterns like T1 and Lackermann's 106 up here. They have very similar killing spectra, and it's also very similar to CYT7 or PTA down here. And so these are not very closely related, but they seem to have the same killing spectra or very closely related killing spectra. So this indicates that there's a high amount of genetic diversity underlying this phenotype within the species. So as I mentioned, my interest really stems from the genetics underlying this phenotype, and so for that, I went in and focused on B728A. It's been a workhorse in the field for some time, And this is also a holdover from my graduate work that I'm most comfortable with this particular strain, and trying to understand what were the genetic determinants for its killing activity. So we have a complete genome. There's online databases that you can feed the genome into, and it'll predict most all of the bacteriocins that are encoded within a given genome. And so it predicted these two different bacteriocins here, and these are just their locus tags, and they're related to E. coli bacteriocins, so they're single proteins, and they have a characteristic catalytic domain and a characteristic immunity protein that protects the producing strain, and so they're very easy to predict. However, when I deleted both of these individually, there was no loss of killing activity. And then when I stacked the deletion, there was still no loss of killing activity. And so this led to the idea that, of course, there's some other source for this killing activity, and where is it at? So doing some literature searches, as well as some more genome gazing, I happened upon another area, and this was just a regulator that I thought might be involved. And lo and behold, deleting it, I abolish all of the killing activity. So what is this doing? Well, it is located in this region that was originally described as a prophage region. And so the regulator I deleted, or the pair of regulators, were here. And then downstream was this annotated prophage region that's actually conserved throughout Pseudomonas syringae. And it's within this larger, or it's flanked by these two, what I use as mile markers, TRPE and TRPG. So it's basically interrupted the tryptophan biosynthetic operon. And so this regulator led to loss of killing activity. And so I had a sense that maybe this prophage region was involved in the killing activity, but of course, this could be regulating many things. Just to remind ourselves, when we're talking about prophages, most people are very familiar with this, but just to make sure, with the bacteriophage, for temperate bacteriophages, we think of them as having these two different life cycles. The characteristic one where it injects its its genome into the host cell, the genome's replicated, the genes are expressed. This leads to formation of new virions that are then released into the environment, and we have virus replication. There's the alternative pathway where once the genome's injected into the host cell, it actually integrates into the host cell genome and is maintained there in a quiescent state. And so it's replicating as long as the cell's replicating, and at some point this will become induced to exit back out and go back into the lytic replication cycle. And so this region that I found looked like an integrated bacteriophage. So as I said, deleted the regulator, and we're anticipating that it's regulating the rest of the genes for the bacteriophage, and then complement it. I also deleted the genes that made up the tail structure of the bacteriophage, as well as the receptor binding proteins, and these are the parts of the bacteriophage that are critical for interacting with its host cell. And then that could be complemented as well. So deletion of the regulator, killing goes away, it's complemented. Deletion of the tail genes also abolishes the killing activity. This was a large region that I didn't want to, it was about 10 kb, and I didn't want to take the time to complement it because it would have been a little bit of a hassle. So then just focusing on the receptor binding proteins, I also lose killing activity, and this is complementable. And so we can say now that this predicted prophage encodes a bacteriophage-derived bacteriocin, or what we commonly just refer to as a tailocin. And so, as Melanie had actually alluded to, her work looking at the Apiaceae of Pseudomonas aeruginosa, these have been studied for a long time, 3-4 decades, if not longer, actually. And since the B728A genome, and there had been a number of syringae genomes that had been sequenced, and we had the aeruginosa genomes, and there had been a number of genomic comparisons, but there had never been a description of a tailocin encoded within the genomes of the strains that I was looking at. It led to this question, why is that? If we've had this information, and we would think that if it was just one co-option of one bacteriophage for this, that we would have seen it in the genome, but that wasn't the case. So again, the question is, are these derived from the same progenitor? So if we look at the genomic sequence, they're actually located in the same area. So here in aeruginosa, just again, the mile markers, TRPE and TRPG, it's the same region for Pseudomonas syringae. But when we compare the nucleotide identity across this region, we can see that the conserved bacterial genes indeed are conserved, right? We think they're vertically inherited, but there's no nucleotide or protein sequence similarity at all between these two Tailocin regions. So it seems the answer seems to be that no, these are derived from independent progenitors. But just to wrap that up, we did some phylogenetic inference comparing these two Tailocins to their progenitor bacteriophages to see if they were clustering similar to each other or with different bacteriophages. And indeed, that's what we saw. So here we see Pseudomonas syringae and all of the isolates up here are most closely related to either phage mu or phage SFV for this particular protein, whereas Pseudomonas aeruginosa is clustering with phage P2. And we see this for all three of the genes that we actually had a reasonable amount of alignment that we could rely on. Most of the other genes that are present, you can't align them, so you can't do these sorts of analyses. Okay, so then the conclusion is these are in fact derived independently of each other. They don't have really any nucleotide or protein sequence identity to speak of. They're clustering with distant bacteriophages. These bacteriophages are related to each other, but people who work on bacteriophages know them to be very distinct from each other. And so this means that there's been multiple levels of convergence that's occurred. First, we have the syntenic convergence, and that both of these Tailocins are ending up in the same genomic region in these two hosts. We also have the phenotypic convergence, where we're going from a bacteriophage to a bacteriocin or to a Tailocin. And so this would also suggest that there's going to be some molecular convergence as well. And what do I mean by that? So just very grossly looking at this structure for the Myoviridae, so these are tail bacteriophages that have a double-stranded DNA genome. You have the head or the capsid. Associated or attached to that is the tail, and then at the distal end again are the tail fibers or the receptor binding proteins. And so this is what it would look like with TEM. When we compare the genes that are conserved here within the Tailocin region for this is B728A back to SFV or phage mu, and just focus on phage mu up here, you can see that only the genes corresponding to the tail region are retained. So all of the genes necessary for regulation, capsid morphogenesis, and such have been lost from the phage mu genome in this Tailocin region. And this was something that had already been understood to occur with the Tailocin for Pseudomonas aeruginosa as well. And so we can see that we have the similar loss of genes between these two systems. And so then you go from bona fide bacteriophage to this structure here, where again, now you can see it's just the tail. There's no head or capsid associated with this structure. So how we think these are working, borrowing from the literature for Pseudomonas aeruginosa. If you look at the outer membrane, right, for gram-negative bacteria, the tail fibers interact with a cell surface receptor on the outer membrane. The tail contracts, drives this core structure through the outer membrane and into the inner membrane, and this is going to deliver the phage genome to the host cell. If you don't have a genome to deliver, though, and we think there's other adaptations as well, but a big part of this is you don't have a genome to deliver, so you're basically an open end up here. And it still is going to contract. It's going to interact or drive the core through the outer membrane and into the inner membrane, but at this point it's going to lead to depolarization rather than phage delivery, or sorry, genome delivery. So as we can imagine with these tail fibers, since it's known from the work with bacteriophages that these are important for interacting with the host cells, this is a great target then for starting to think about the specificity between a given Tailocin and the cells that are, or the strains that are targeted by that Tailocin. And so if we look at this region across a number of different strains, we can see that the receptor binding protein itself tends to be the most divergent. And so this region here of sequence similarity would extend for about 10 or 15 kilobases upstream, which are all the core components of the Tailocin. But with the receptor binding protein and its associated chaperone, there seems to be a lot of evolution, recombination that's occurring in this region. So can we engineer retargeting in this way? And the answer is yes. So for PJA, pathovar japonica, it is not natively able to kill pathovar actinidia or pathovar glycinia, as I'm showing up here. However, if I just take those two genes, the receptor binding protein and the associated chaperone, from B728A and express it in japonica without anything else, I can now retarget it to kill these two strains because B728A natively kills those two strains. And so I've done this for a number of different strains, and all of this activity is transferable from PJA. I've done it with B728A as well, and then these other strains. So basically everywhere where I've looked, I don't anticipate all of this activity is derived from the Tailocin, but it seems like that's the majority of the killing activity. Because I have some evidence, and I'll show you that in a second, of what some of these other bacteriocins may be doing. And again, these killing spectra are readily modifiable. Okay, so now shifting to, we have essentially the key in this scenario, right? So what's the lock? What is it about the host cell that's also determining the specificity? We focused in early on lipopolysaccharide, and we had a couple of reasons for doing this. One, all of the bacteriophages that these Tailocins are related to, they all target LPS. And most people are familiar with this. It's a major component of the gram-negative outer membrane, where you've got this lipid portion here that's embedded in the outer membrane, another portion of the core oligosaccharide that does vary a little bit, but tends not to vary very significantly among strains, and then the highly divergent O-specific or the O-antigen component that's highly variable among strains, even within a species. So as I mentioned, this is a known receptor for the bacteriophages that the Tailocins are derived from, but also, as you would expect, for the Pseudomonas aeruginosa Tailocin as well. And so we received a number of strains from Matt Templeton's group in New Zealand where he had characterized LPS mutants. And so these are all transposon insertion mutants. So I'm showing up here at the top, we have wild type LPS. This is the wild type strain. And then these are two strains that have a transposon insertion, but it's not associated with LPS. And so we can see that the killing activity for this strain is Tailocin derived. Because the structural mutant and the receptor binding protein mutant, they lose all of their killing activity. We still have killing for these LPS mutants down here for the wild type, and we would expect that if the LPS is the receptor, that we wouldn't have seen any killing activity. However, there's two things that you can see right away. First is that these killing zones are phenotypically distinguishable from these. So these have a very sharp edge, which is characteristic of a Tailocin killing because it's a big, bulky structure that cannot diffuse efficiently through the agar. Whereas for other compounds like smaller single protein bacteriocins, they're much smaller, and so they can diffuse through the agar matrix, and so you can get this diffuse killing zone here. But the other bit of evidence that showed that it's clearly not the Tailocin is in the Tailocin deletion mutants, we still had the same killing activity recovered. And so this indicates that there's actually some other source of the killing. And so, as I had shown before, the deletion of this particular bacteriocin doesn't affect the killing of the wild type strains with the wild type LPS, deletion, complement, and then the mock complement. However, now with these LPS mutants, we can see that by deleting this alternative bacteriocin, all of the killing activity is abolished, and this is all complementable as well. So we have a genetic distinction here between the killing activities, but we can also distinguish them physically as well. So by doing a step of PEG precipitation, it's just a simple method where you can separate bulky, high molecular weight molecules from smaller molecular weight molecules. This is very efficient for recovering, and it was developed as a technique in the bacteriophage field. You can see that we can recover all of the Tailocin-mediated killing activity, the two mutants, complemented mutant, and then a mock complement. But all of the killing activity against the LPS mutants was completely lost and not recoverable. And so this is really interesting and is something actually that, to my knowledge, has not been reported on within the bacteriocin field, and that's that gaining resistance to one Tailocin led to sensitivity of the other Tailocin. So it's not only is there redundancy here, but it's conditional redundancy. It's not that they're both killing at the same time, but one has to be sensitive, or sorry, the strain has to become resistant to one to become sensitive to the other. So additionally, we recovered a number of strains that were resistant, de novo resistance to the Tailocin. And so working here with the green being pathogen, Pathovar phaseolicola, as well as the soy pathogen, Pathovar glycinia, just essentially exposed these cultures to the B728A Tailocin, selected for resistant colonies, confirmed their resistance, and sequenced their genomes. And as we would expect, most of the genes that we recovered, this is just a small subset of the mutants that I've sequenced, they all map back to genes, glycosyl transferases in particular, and these are well known to be involved in LPS biosynthesis, and they're closely related to those that have been described in E. coli. And so the question that I had coming out of this, thinking about how these interactions again between these microbes in the environment, we expect that there will be selection for resistance that will be imposed within the environment. And so if a strain does become resistant to a given bacteriocin, in this case to the Tailocin, does that affect its ability to interact with its host plant? And so for both of these strains, the answer was yes. Here we're looking at just infiltration, in this case, the green bean pathogen and green bean. The black bar here is the wild type. The gray bar here is Harpel deletion, so this is blocked in its ability to express the type 3 secretion system. And then all of these other strains here, BRM is bacteriocin resistant mutant. These are all independent isolates that have independent mutations. And we can see that after 24 hours, they really have a growth defect, except for this last one, 11. It has something of a growth defect, but seems to do better than the other strains. And that this growth defect does persist at 48 and 72, at least for some of the strains. I expect that this is actually true for more of these strains, that there was just not enough replicates here to get the statistics to work. And the same is true for Pathovar glycinia. So we can see again, at 24 hours, these strains are doing just about as poorly as the Harpel deletion mutant does, although they do significantly better at 48 hours and 72 hours post-infiltration. And again, I think there is some replication issue here that, but I've seen this multiple times with this experiment, multiple independent experiments. They always lag behind the wild type strain, even at these later time points. Okay. So we know some of the genetics underlying the bacteriocin production, as well as what the determinants for the sensitivity are. And so now we want to ask, well, does this actually matter in an ecological sense? And so again, going back to some of the literature from the mid-60s, so for example, a study here by Garrett and colleagues, where they were looking at pseudomonads that they were taking off of different fruit trees. And this is a little busy, but I just want you to focus on here when they looked at the bacteriocins they were producing. And the majority of isolates coming from pear didn't kill other isolates from pear, but they did kill isolates coming from citrus. And the converse was also true, that the majority of strains isolated from citrus don't kill other citrus isolates, but do kill pear isolates. And so I had the opportunity to collaborate with Carolee Bull when she was still at the USDA in the Salinas Valley to ask some more questions based on collections of strains she had had. So many people may be familiar with the Salinas Valley. It's essentially the salad bowl of America during the spring and summer growing seasons before it shifts largely to southern Arizona for the winter growing. And what she had noticed in this valley, there had been two different pathovars. Pathovar Apiaceae that would cause leaf spot on celery, and a different pathovar, Coriandricola, that would cause leaf spot on cilantro. And so these have been known to occur within the Salinas Valley. And in the late 90s, early 2000s, there was this new disease that emerged, leaf spot of parsley. So these are all in the Apiaceae family. But the interesting observation that she had is that within a field or within a given plant, even though both of these pathovars exist and are causing disease within the valley, she never found a mixed infection. So this would either be caused by Pathovar Apiaceae or pathovar Coriandricula, but she never found them co-infecting. And so our question was, well, is this potentially bacteriocin-mediated inhibition that's leading to this difference? And so we screened a number of API isolates and related strains and compared those to the Coriandricula isolates. And so now I'm just lumping everything together. I'm not showing every isolate individually, but what I want to draw your attention to is that essentially all of the API isolates killed all of the Coriandricula isolates, right? And so this doesn't guarantee that this is why there's this trade-off going in the field, but it's a strong indication that likely this is why you're only getting one or the other infecting, even though they're both competent in infecting this host. And so, you know, originally I thought maybe there was going to be reciprocal antagonism, but it turns out that that's likely not the case, that it's actually the pathovar API. If it encounters the host first, then it's going to block Coriandricula, and Coriandricula is only going to infect in the absence of API. So of course there's work that we're planning to do where we'll get some resistant mutants to Coriandricula and then see if now we can get a mixed infection. And then conversely, go in and delete the bacteriocins out of API and see if that also will allow for the same co-infection to occur as well. And so that's ongoing work. So with that, I would just like to acknowledge my collaborators on this work, my postdoctoral advisor, Dave Baltris, as well, a collaborator, Tanya Renner, who really helped out with the molecular phylogenetics for understanding the evolution of the Tailocin, or the derivation of the Tailocin from independent bacteriophages. And then Carol Liebel, again, she has now moved to Penn State. And then I've had a number of students that have worked with me and contributed to the work that I've shown. And then, of course, I've gotten support through the SDA for this work. And so with that, I'll be happy to take any questions.
Do the students have any questions? First, we'll go with the students.
Andy's not here, so someone has to ask one. With the two parsley pathogens, do you see that there's like more API caused like outbreaks because it's theoretically outcompeting the other?
No, that's a really good question, and that's something that I would really need to talk to Carol Lee about because she has that data. I don't know if that's true, but that would be an expectation if it's inhibition in that way, unless there was some other compensatory thing that API is not as good at remaining in certain reservoirs or something like that. There may be some trade-off there, but we would expect if that's the only thing going on that API may become the dominant one. Yeah, good question.
A question. I believe earlier in the talk you said that in order for one of the Pseudomonas syringae isolates to be resistant to one bacteriocin, they must there was some trade-off in susceptibility to a different one. And I was wondering if you could explain why that might be, or are there genes that are specifically being traded in and out at the same locus that might cause that?
So, sorry, just to reiterate, you're asking for the trade-off in the sensitivity between the two. What is the basis? Why is there that trade-off? Yeah, and the answer is I don't know right now. I have a couple of hypotheses. So LPS is well known to serve a protective function. And so the idea may be, one, it's the largest component of the outer membrane. And so potentially by losing the LPS in its full structure, there's still, the lipid A is going to be there, probably the core oligosaccharide may still be there as well. But it's lost all of the OA antigen. And the thought is maybe there's some stress in the outer membrane that's leading to upregulation of an alternative cell surface receptor. That cell surface receptor, or sort of similar but different hypothesis, is that that receptor is always there, but the other bacteriocin, one of the function of the LPS, again, we think about it protecting against oxidative stress, antimicrobial peptides, but it may also be protecting this alternative receptor from this other bacteriocin being able to access it. And so there's a couple of different questions. But other than just the loss of the disruption of those LPS genes, there shouldn't be anything else different about those strains. Complementing them and showing that it goes back, I think, would be critical to showing that. Yeah, but it's a good question.
So I'm just wondering, so what's the expression pattern about the prophage
Yeah.
Is it inducible expressed or just consecutive expressed? So,
Yeah. No, it's a really interesting question. When is this going to turn on? Potentially within the plant environment, right? That's where we would care about. So I do know that after infiltrating a cell into a susceptible host and coming back 24 hours, I can recover the specific killing activity. So they are getting induced once they're inside. I anticipate that potentially certain stressors on the leaf surface, like UV exposure or desiccation stress or starvation, will also induce them. So for all of mine, right, I use mitomycin C. It's sort of the sledgehammer to get them to turn on all of their SOS systems and produce all of these different compounds. The interesting thing about this is it couldn't be constitutive because just like with the virus, it's too bulky to be exported out of the cell. And so there's only a subset of the cells at any given time that are going to express this because they essentially lyse to release these into the environment. At least that's what our hypothesis is based on all of the previous research that's been done. And so looking at the dynamics of induction within the plant environment, I think is something I'm very keen on pursuing. Yeah, good question.
Any more questions?
Is the producing strain sensitive to its own tailocins?
Mm-mm. And so if you saw there, it was all black boxes on the diagonal there. And so there would obviously be a high degree of selective pressure to become resistant if you have been sensitive. And so there's two things related to that. For the small, like the 4651, that small single protein bacteriocin, we know that the producing strain also has a characteristic immunity protein associated with it. For the tailocins, it's probably just that its LPS is not recognized by the tailocin as it's produced. But there's been some work looking at evolution in biofilms and Pseudomonas aeruginosa, and you will get these sweeps where a strain will change its LPS to them suddenly become sensitive, and they can show it starts to kill itself, and so then it quickly has another mutation that blocks that from occurring. So I think that does crop up in nature, but it's quickly stamped out.
Okay. Oh, no.
So let me make sure I understand it. So with your initial screen, you're using mitomycin C-induced donors to get extracts that are then tested in various lines and then used. I mean, do you get different patterns of interaction if you look at competition in leaves as opposed to this mitomycin C-induced assay?
No, and that's something that I definitely am interested in looking at, is starting to move back into the plant environment, but we haven't looked at co-competing them within a leaf environment to see how much it actually matters, because you may still just get, based on where you find them distributed, they may be able to coexist as long as they're in different patches that are isolated. But over enough times of, within a given field, potentially of interacting enough, you might see it sweep from one to the other. But it's a good question. Yeah.
Okay. Don't have any more questions? Thank you.
This has been a production of Cornell University, on the web at cornell.edu.